Alright, let's get back to metabolic tasks. The next metabolic task is biosynthesis. Biosynthesis uses precursor metabolites, which are made by catabolism, to make what we call building blocks. Those are small molecules, very specific molecules for each cell. Building blocks then will be used in next metabolic task to make macromolecules. We call them polymers. For your exam, you have to know samples of monomers and polymers, and also you have to know the difference between monomers and polymers. So, monomers or building blocks, those are small molecules. If we connect them to each other in certain way, in certain order, we will form polymers. Example, let's say proteins. Proteins are polymers. They are large molecules. Proteins are made of amino acids, which are monomers, building blocks. Polysaccharides are polymers, macromolecules, are made of simple sugars, which are monomers, for example, glucose, maltose, sucrose. Next example, peptidoglycan. Remember, all cell walls of bacteria are made of peptidoglycan. You already know it is a polymer, which is made of monomers NAG and NAM. And the last example, lipids. Lipids are polymers made of monomers, fatty acids. Polymerization is the next metabolic task. In this step, building blocks made by biosynthesis are joined together to form macromolecules or polymers. For example, proteins, DNA, RNA molecules, peptidoglycan, polysaccharides. In order to form polymers, building blocks must be joined in a specific order. For example, structure of protein is determined by the order in which its amino acids are joined together. So, how cells know which amino acids in which order they have to connect in order to form certain specific protein? All this information is written down into chromosome, into DNA molecule. And the final step of metabolism is assembly. This is when parts of the cell are made. Please remember, some parts of bacterial cell, for example, flagella, can be made by what we call self-assembly or spontaneous assembly. What does it mean? It means if you take flagellants, proteins, and place them into laboratory tube, add some primers, they will spontaneously assemble into flagella. Even more complex cellular structures, such as ribosomes, assemble spontaneously. But formation of, for example, cell wall is example of assembly that is catalyzed by enzymes. Means when cell wall is formed, it requires additional chemical reactions. Alright, so far we've been talking about aerobic metabolism. Now let's compare aerobic metabolism to anaerobic metabolism. Let's talk how anaerobic bacteria make ATP molecules. Same as aerobic bacteria, anaerobes use two ways to make ATPs. The first one is chemiosmosis, and remember the main difference between aerobic chemiosmosis and anaerobic chemiosmosis in terminal electron acceptor. For aerobes, terminal electron acceptor is always oxygen. For anaerobes, terminal electron acceptor is something else, sulfates, fumarates, nitrates. But what is the second way that anaerobic bacteria use to make ATPs? The second way is called fermentation. And same as substrate level phosphorylation, fermentation entirely depends on the substrate. And please remember, the substrate for fermentation is always sugars. There are different types of fermentation. For example, lactic acid fermentation, ethanol or alcoholic fermentation, and mixed acids fermentation. All those different types of fermentation use the same substrates, sugars. What is the end product of fermentation? The end product of fermentation is acid, for example, lactic acid fermentation, or mixture of acids, for example, mixed acids fermentation. And some types of fermentation also produce gas, for example, CO2 or hydrogen. On this slide, you see picture formulas of lactic acid fermentation. So as you see, the substrate for lactic acid fermentation will be used at glucose, and then there will be chain of biochemical reactions, and the end product of lactic acid fermentation will be two molecules of lactic acid. Bacteria can preform lactic acid fermentation to make ATPs, because you see two ATPs will be made by these lactic acid fermentation reactions. But keep in mind, humans' tissues also can perform lactic acid fermentation, and that is a muscle tissue. When muscle tissue deprives oxygen, let's say we work out very hard, then muscle tissue start performing lactic acid fermentation to produce additional ATP molecules as a source of energy. Remember, the end product of lactic acid fermentation is lactic acid. That explains why next morning we wake up and feel pain in our muscles, because a lot of lactic acid will be deposited there. This slide shows you that different microorganisms perform different types of fermentation. For example, Streptococcus species, Lactobacillus species, Bacillus species can perform lactic acid fermentation. So the end product of this type of fermentation will be lactic acid. Yeasts can perform ethanol or alcoholic fermentation, and the end product will be acid, ethanol, and carbon dioxide gas. Propionibacterium species perform mixed acids fermentation. So as you see, the end product will be mixture of acids and gas. And then you see Clostridium species, Escherichia species, Salmonella, Enterobacter species, they all can perform different types of mixed acids fermentation. And you see the end products for each type will be mixture of acids and gas. So how can we use our knowledge about fermentation in our microbiology everyday life? In our labs, microbiology labs, we use what we call fermentation tubes tests to identify microorganisms from humans' gastrointestinal tract. On this slide, you see a picture of fermentation tubes. Different fermentation tubes contain different sugars. Remember, the substrate for fermentation is sugar, so we take different simple sugars, sucrose, lactose, maltose, glucose, to make different fermentation tubes. Initial color of fermentation tube is pink or red, like in the picture A, tube A. Inside of each fermentation tube there is a small tube that is placed upside down, and we call it a Durham tube. We use Durham tube to capture gas if it's produced as a result of fermentation. In order to identify anaerobic bacterium from gastrointestinal tract of our patient, we take different fermentation tubes with different sugars and inoculate each of those tubes with isolated bacterium that we would like to identify. Then we incubate fermentation tubes for about 24 hours, and in 24 hours we pull them out of incubator and identify result. When we evaluate results of fermentation tubes, we always answer three main questions. First question: is there bacterial growth in the tube? If there is a bacterial growth in the tube, you will be able to see cloudness in the tube. Second question: do we have fermentation occurred in the tube? If fermentation occurred, the color of the tube will be changed from pink into either orange or yellow. And the last question is the gas was produced. If gas was produced as a result of fermentation, you have to check Durham tube. If gas is produced, you see gas bubble in the Durham tube. If you don't see gas bubble in the Durham tube, means gas wasn't produced.